Clarity Clinical Solutions.
Orphan drugs and rare disease trials. Hope for millions. How incentives, innovation, and determination are bringing treatments to patients with rare diseases. In rare disease research, patient advocacy groups are often the driving force. They fund early-stage research and proof-of-concept studies, create and maintain patient registries, connect patients with researchers, educate the community about clinical trials, and lobby for policy changes. Notable examples include the Cystic Fibrosis Foundation, the ALS Association, the National Organization for Rare Disorders, the Every Life Foundation, and Global Genes. Many rare diseases are caused by a single gene mutation, making them ideal candidates for gene therapy. Examples include Zolgensma for spinal muscular atrophy, Luxturna for inherited retinal disease, CAR-T therapies for rare cancers, and antisense oligonucleotides for specific mutations. However, challenges include extremely high costs, often over $1 million, delivery to target tissues, unknown long-term durability, manufacturing complexity at small scale, and insurance reimbursement barriers. Economic and ethical dimensions. Balancing innovation incentives with patient access. Orphan drugs are among the most expensive treatments in medicine. The average orphan drug costs over $150,000 per year, and gene therapies can cost over $1 million for a one-time treatment. The small patient base means high per-patient research and development costs. Ethical questions arise around whether prices are justified by R&D costs, how to ensure equitable access, whether profits should be capped for taxpayer-funded research, and how to balance incentives with affordability. For the millions living with rare diseases, the past 4 decades have brought unprecedented progress, from fewer than 10 treatments to over 800. With continued innovation in trial design, gene therapy, and patient-centered research, the next decade promises even more breakthroughs for the rare disease community. Thank you for watching. Brought to you by Clarity Clinical Solutions, LLC. In the United States, a rare disease is defined as one affecting fewer than 200,000 people. In Europe, the threshold is fewer than 1 in 2,000 people. There are over 7,000 known rare diseases, about 80% have genetic origins, and approximately 50% affect children. Yet only 5% have FDA-approved treatments. In total, 30 million Americans and 400 million people worldwide live with a rare disease. The Orphan Drug Act. A landmark law that changed the landscape of rare disease treatment. Before 1983, only a handful of treatments existed for rare diseases. The pharmaceutical industry had little incentive to develop drugs for small patient populations. The Orphan Drug Act changed everything. Key incentives include 7 years of market exclusivity, tax credits for 25% of clinical costs, FDA assistance with trial design, grant funding, and waived user fees. The impact has been dramatic. From fewer than 10 orphan products before 1983, we now have over 800 approved orphan drugs. Running a clinical trial for a rare disease is fundamentally different from traditional drug development. Patient recruitment is extremely difficult. Populations are very small, patients are scattered globally, many are undiagnosed or misdiagnosed, and pediatric cases add complexity. Scientific hurdles include limited natural history data, difficulty defining endpoints, heterogeneous disease presentation, and a lack of validated biomarkers. Innovative trial designs. Creative approaches to generate evidence with very small populations. Traditional large randomized trials are often impossible for rare diseases. Researchers have developed creative alternatives including N of 1 trials, sequential multiple assignment designs, Bayesian adaptive designs, and enrichment designs for genetic subtypes. Regulators offer flexibility too. The FDA accepts single-arm trials, external or historical control arms, surrogate endpoints for accelerated approval, and post-marketing commitments for confirmation. Understanding the natural progression of a rare disease is critical for designing successful trials. Without understanding how a disease progresses naturally, you cannot design endpoints that detect meaningful improvement. Natural history studies track patients over time to establish benchmarks, identify the best outcome measures, and often serve as the control group for single-arm trials. Patient registries collect longitudinal data, help identify eligible patients, build community trust, and are preferred by regulatory agencies. Patient community. How advocacy groups and patient communities drive rare disease research.